be taking questions. We're going to be taking questions through the chat feature you can see on the bottom of your screen. And then Gina's going to moderate, and you can enter your questions through the chat, and basically we'll try to answer them as quickly as we can. And just want to let you know one quick thing. 650 people have entered into our Zoom room in the month of January. So, which is pretty amazing on our weekly events and our Sunday talks. So I'm going to pass it over to Bill, our president, Bill Westlake.
Hey everyone, I'm so glad to see you guys tonight. I just wanted to give an update like we typically do on the ALK Talks. This one is a very pertinent update, especially with Alice Shaw being on tonight. First off, 4.5 years, Lectinib first line for me. I live in Atlanta, Georgia, on the board, currently serving as president. That's a big honor for me to help serve. Sometimes we do polls on the ALK Talks at the very beginning. If we did a poll, and let's just say that poll were this: do you consider your oncologist to be an ALK specialist, like an ALK expert? Now, if we did that poll, especially on this call, I would expect that a majority would say no. And we're a rare disease. So it's whether you're in a major metropolitan city, a small community, it's likely that you have a good oncologist. If you have one, they're not necessarily an ALK expert. So why I bring this up is that ALK Positive introduced a couple of years ago, it's called the Second Opinion Program. And I like to announce this program, especially to this audience, because you may not know it exists. It was founded and funded by Jim Peard, who came out of the ALK Positive support group, and later additionally funded by Kelly Culliver from the Culliver Foundation in honor of her husband, Rick. And the program literally connects patients with ALK Positive cancer worldwide to an ALK specialist. So in 2018, we had, during the launch, we might have had 3 specialists, and Alice Shaw was one of them. Now the program covers 6 states. We have 9 to 10 specialists, and we hope to even double that. We don't want to overload our ALK specialists out there. So it's really important that if you're in a situation where you have progression, it's really a good idea to get a second opinion. The other situation you might be in is you're not sure if your treatment is that effective or not. And you question that. So rather than stress over that and just continue to work with your existing oncologist and their team, consider going to the ALK Positive website and reviewing the application and submit your name for a second opinion. And you will receive then the ALK specialists around the country that you will have to go visit under most conditions. And in one condition, you can do a remote consultation, and ALK Positive will pay for that appointment. Even if you have insurance, you can apply your insurance, and whatever's net of that, ALK Positive will pay for it. So that's really, really critical. That's really it. So just know that it's out there, and grateful for everyone's participation with ALK Positive Inc. Thank you very much.
Thank you so much, Bill. We're so lucky to have that. And I'm so excited tonight to be able to introduce the Queen of Alk. And I think she was originally dubbed that by one of our heroes, Linnea. And so Dr. Shaw was in practice at Mass General for a long time. And I was lucky enough to actually come and see her. I felt like I was seeing rock stars there the whole time. But I wanted to just kind of hand it over. For those of you who don't know her that well, I want you to get a chance to meet her and know how amazing she is. So Dr. Shaw, will you just tell us about your career a little bit and tell us where you are now, what you're doing?
Well, thank you so much, Gina, for such kind words. And it's great to see many of you. I see many familiar faces, many familiar names. I don't see all the faces, but it's just great to be here. Gina has been really, really kind to actually pose a number of questions to me, one of which she just asked. And I thought today, and I know you guys have heard a lot of presentations and talks from a lot of the researchers. And I thought today that it'd be nice to maybe make it a little more informal, and also just give lots of time for Q&A. Because when I was last here, there were a lot of questions and I felt like we couldn't get through to everything. So this will hopefully be a lot more interactive. But I'll start with Gina's first question, which was about sort of a little bit about my career and what I've been doing. I'm from Maryland originally, and then after high school, I came up here to Boston, and I've been in Boston since then, here at college and medical school, graduate school, and all my medical training. And I did a lot of research up here, actually, basic research, first studying sort of basic transcriptional regulation of blood cell development. That was when I was a grad student. And then as a postdoc, I was at MIT with Tyler Jack studying actually the KRAS oncogene. But around 2007, I decided I really wanted to be closer to patient care. And so at that point, I transitioned from a basic investigator at MGH and MIT to a translational and clinical investigator at Mass General. And somewhat fortuitously, ALK was actually discovered by a team in Japan when I first started at MGH as a clinical investigator. And it was one of those like aha moments, because we could tell even just from that very first study that was published in Nature that ALK was going to be a really big target. And at that time, many of you don't realize, but at that time, we really had very little in the lung cancer space. We were just learning about EGFR as a target, and that was really it. And so in 2007, ALK came along, and I think it really kickstarted the whole field of precision oncology. Now, some of you may also know some of the history of ALK and how there was also serendipity with ALK, because when ALK was first discovered in 2007, we were already testing crizotinib, which is the first ALK inhibitor, in the clinic. And again, some of you know this story, but for those of you who don't, crizotinib was actually being developed for a different target called MET. But we did know that it inhibited ALK. And so when the first pivotal study of ALK was published in 2007, we started working very closely with John Iafrate, who I think you guys met last year, to begin to diagnose patients with ALK. We also worked very closely with Jeff Clark and Eunice Kwok, who were running the phase 1 trial. And we basically could pivot very quickly and start enrolling patients with ALK onto the phase 1 trial of crizotinib. And I remember so clearly the very first ALK patient who we treated with crizotinib, and he had started, he was a patient of Tom Lynch's, but I knew him well from the trial, and he had started on crizotinib the day after Christmas in 2007. So think about that, 14 years ago. And within days, he was feeling better. And within a few weeks, I believe he actually had even been able to return to skiing. And it was just like this incredible transformation. And so at that point, seeing this very first ALK patient and his response in 2008, I think many of us knew that ALK was, and actually ALK and the ALK targeted therapies like crizotinib were really going to be transformative for patients. And so soon after that first patient, we started enrolling more ALK patients. Linnea was one of the first ALK patients as well, who we treated on the phase 1 crizotinib study. I think many of you know her or know of her. I actually have a number of patients who we started on the phase 1 crizotinib study back in 2008 and 2009, who I still follow today on crizotinib, commercial crizotinib, of course. So that is pretty incredible. And I think, I think all of that just really highlighted to us that ALK positive cancers just have this incredible, remarkable sensitivity to ALK targeted therapies. And again, just to remind you, back then, all we had were EGFR targeted therapies. And so this was really a huge development at that time. But unfortunately, even as amazing as these responses were, we actually started to see fairly early on that many of our patients on crizotinib were developing resistance and actually relapsing even as early as, within the first year. And so it became very clear that to continue to advance the field, we really needed to understand what was driving resistance. And so I set out working with a number of my colleagues at MGH, including Jeff Engelman and Justin Gaynor, to study resistance, both by modeling it in the lab, and also, I think more importantly, by studying biopsy, cancer specimens from patients. And so for about a decade, we studied resistance intensely. And in parallel, we also teamed up with various biotech and pharma to develop better ALK inhibitors. And early on, even we were starting to identify resistance mutations to crizotinib, for which second generation inhibitors like ceritinib or Zycadia, alectinib, brigatinib were very, very active in preclinical studies. And so all of our work was able to really set the stage for testing these second generation agents in patients who had become resistant to crizotinib. And then soon after, we moved these agents into the first line setting where we know that they have even greater impact. But again, even with second generation inhibitors, we saw this problem of resistance rearing up again. And so again, launched many different studies to understand resistance to second generation drugs, started working on a third generation drug, lorlatinib, with Pfizer, which had been developed specifically to overcome resistance and designed specifically to penetrate into the brain to address the problem of brain metastases. And soon we were able to bring lorlatinib into the clinic, show activity after first and second generation ALK inhibitors. And I think more recently, what's been important to show that lorlatinib is particularly effective when given as first-line therapy. And likely this is because lorlatinib can really block all of the known single ALK resistance mutations and can also not even just treat brain metastases, but protect the brain from metastases. Now, about 2.5 years ago, we were continuing to push the boundaries, I think, for ALK, and we were actually already well beyond lorlatinib at that point. We were planning combinations. We had lots of basic research into how ALK positive cancers evolve over time, why it is that some rare ALK positive cancer cells persist or survive despite being exposed to an ALK targeted therapy. How ALK positive cancers rewire and reactivate other pathways to cause resistance. We also were studying why ALK positive cancers don't respond to, drugs like Keytruda or Opdivo, these so-called checkpoint inhibitors. And we were also laying the groundwork for this new fourth generation class of ALK targeted therapies, which we'll come back to. But it was clear to me at that time that, as an investigator and having treated so many hundreds and hundreds of ALK patients, that, we really needed to go beyond the current toolbox of targeted therapies and really think outside the box, because it really felt like we needed to continue to have a rich supply of treatment options for patients. And around this time, as I was coming to that realization, my former colleague and friend, Jeff Engelman, had actually moved to Novartis to lead the oncology R&D, research and development at Novartis. And he actually ended up recruiting me to Novartis to work with him and to lead early clinical development. Initially, I was actually I was quite hesitant to go, in large part because of all of you, my patients. But I also realized that it would be an incredible opportunity to apply what I had learned and, led in the ALK space so that I could help other patients in need with different types of cancer. But also, I realized it was an opportunity to learn about all of the innovation going on outside the ALK space in order to bring some of those learnings back to ALK, hopefully in the form of new and different therapeutic approaches. And so 2 years ago, I transitioned myself to NIBR. This Novartis Institutes of Biomedical Research. It's just right across the river from MGH, about a quick 15-minute ride. And I currently lead a group of about 200 researchers, clinical clinicians and researchers. And we develop and design and execute phase 1 first in human trials of all different types of therapies, targeted therapies, as well as immunotherapies, CAR T therapies, even these new so-called radioligand therapies. And we also treat all different types of cancers. Some of you know that I've been able to maintain my clinic at MGH still. It's very small, but I do come back every Thursday afternoon, and I have a small panel of patients, some ALK and ROS patients, and a few others, and also been really happy to be able to maintain my work with my former colleagues like Justin, Jess, and Ibi. And I know they've been doing amazing work continuing to push all of the ALK research forward. So maybe, Gina, that was a long answer to your first question, but do you want to pause here and maybe take some questions?
Yeah, let's pause and give you a chance to breathe too. That was amazing. And I think we're probably just going to absorb every single word that you say. So we appreciate you so much. But yes, take a break. And
I'm good. I'm good.
Okay. Our next question we're going to ask is really about ALK. So, a lot of times with ALK, we talk about brain progression and things like that. But could you tell us a little bit about how ALK is different when it comes to bone progression and like how you treat patients who have bone progression?
Yeah, so you're right. We often speak more about sort of progression in the brain because it is actually, we had shown previously that for some reason, ALK positive lung cancers have a propensity to spread to brain. Fortunately, that is less of an issue now that we have such brain penetrant drugs like alectinib, brigatinib, and lorlatinib, although of course we still face this problem. But the truth about ALK is that it does tend to disseminate or metastasize early. And oftentimes it's not just the brain, but to other organs. And bone is one of those organs where ALK positive cancer can spread to. And I have had some ALK positive patients over the years where for whatever reason, we don't understand why this is, but their progression tended to be primarily, sometimes only in the bone. And this happens for other patients with different types of lung cancer as well. Their cancer has, for some reason, this tropism to grow in the bone. Now, how we manage that is typically through continuing to target ALK itself, because of course the cancer that's growing in the bone is still the same ALK positive lung cancer. It may have acquired a resistance mechanism. So, if we, for example, have a patient on alectinib and they have now primarily progression in the bone, and we do, for example, a liquid biopsy, so we sample and sequence the circulating tumor DNA, and we find a mutation like G1202R, we're going to move on to a drug like lorlatinib, which should work everywhere, including the bone. But there are other bone-specific things we do think about. So, for example, sometimes we will need to use radiation, so local therapy like radiation, because a patient may be experiencing pain at the site of progression in a bone, and the radiation can be very effective at treating the pain. Sometimes, especially if there's been a fracture or there looks like there's going to be an impending fracture, we may even have to involve an orthopedic surgeon, and so actually go to surgery and stabilize that bone. The other thing I would say about bone metastases is that when a patient has progressing bone metastases, we do want to make sure their bones are as strong as possible. So using drugs, what we call bisphosphonate drugs like zoledronic acid or Zometa, it's very important and has been shown. Those types of drugs have been shown to decrease the risk of skeletal events like fractures. So those are important alternative. I wouldn't say alternative, but they're important treatments to be aware of besides the therapy for systemic disease, such as another ALK targeted therapy, or maybe it's chemotherapy or a trial. Also, radiation could be indicated, surgery could be needed, and then of course, keeping the bone strong, very important to keep in mind.
Okay, awesome. There have been some questions just kind of about therapy and how you handle that. One of our friends in Germany said, there's not always the same options available internationally as there are in the United States. And in the United States, sometimes maybe doctors even take a little bit of a risk in trying things that they wouldn't do because there's not actually published data on trying these things. And one of those things was dose escalation. So increasing your dose. Would you ever say that someone could start on a low dose of a TKI and work their way up according to progression? And if they reach the maximum dose, would it be okay to increase past that just to see if it would work?
Yeah, so you're really getting at how we may consider personalizing even the dose of a targeted therapy based on an individual and their own sort of medical condition. And we do that to some extent. I would say that all the medicines that we use for ALK, right, the approved drugs like crizotinib, ceritinib, alectinib, brigatinib, lorlatinib, they're all approved at a so-called standard dose. And that means that that's the dose that was established in clinical trials to basically be a standard dose based on looking at the safety, looking at the exposures of the drug that were reached in patients, oftentimes based on the pharmacodynamic effects, meaning that we were able to show that ALK was fully inhibited, and of course, based on efficacy, how active was the drug. And usually we hope that standard dose really represents the best balance of safety and efficacy. But what we've seen over the years is that it's hard to find the right dose because everyone's different. And so just as an example, ceritinib was one of our first second generation inhibitors approved. It's not used very much anymore, but it's a good, I would say, learning case, because the dose that was selected, again, you had to go back in time when we were just developing ceritinib. At that time, we had really very few other options for patients failing on crizotinib. So there was a huge rush to get this drug available to patients. And so I think in sort of the rush of developing ceritinib, I think that the sponsor, the drug maker, additional studies could have been done to further sort of hone in on the right dose, but they're really eager. Patients were everywhere around the world clamoring to get access to this new drug. So they chose a dose, 750 milligrams once daily fasting, that I think most of the investigators who worked in that trial knew it was too high of a dose. And so even though that became the standard dose, I think many of us were concerned because very few patients could tolerate that standard dose. And so in the end, actually what happens is that most patients, if they even started the standard dose, would definitely have to dose reduce. And those of us who worked with that drug knew that they would need to be dose reduced. So we started treating at a lower dose. And so there we already were making some modifications based on our own experience. And I would say that's played out for many of the other ALK inhibitors, especially, for example, lorlatinib. Many of you who may be on lorlatinib, the standard dose is 100 milligrams once daily. Again, many of us who worked on the phase 1 trial felt that was going to be too high of a dose. And in fact, a higher, probably the needed dose was chosen because the sponsor was really hopeful that we could also inhibit one of the resistance mutations in ROS. But for ALK, lorlatinib was incredibly potent, and even 75 milligrams was as potent, it appeared, as 100 milligrams, yet the standard dose was 100 milligrams. So again, I think lorlatinib is a good example where probably the standard dose is a little too high for most patients. And so for me, I routinely start patients at 75 milligrams of lorlatinib rather than 100. Now, what about starting even lower, Gina? Because you were saying start low and then bring it up according to what's going on with progression. That we don't do, because if you start really low, like almost kind of sub-therapeutic levels, then the worry is that you could very gradually cultivate resistance because the cancer cells aren't being exposed to really therapeutic or highly efficacious doses. So when we dose reduce, I would say if I start at a slightly lower dose, it's still at a very active dose that we've shown in the clinical trials is very active, and patients will have almost certainly the same outcome as if they started at that slightly higher dose. Now so I don't start too low, Gina, but I start oftentimes one dose level down based on sort of experience in the clinical trials and clinical experience overall. Now we do the alternative, which is to dose escalate in some cases, particularly with drugs like alectinib. Alectinib, as many of you know, I think the survey I heard about 60% of you may be on alectinib. So many of you are familiar with alectinib. It's very well tolerated at the standard dose of 600 milligrams twice daily. But at 600 milligrams twice daily, it covers the body really, really well. It also covers the brain, but not quite as well. And so many years ago, we had seen that we had a number of patients on the standard dose of alectinib who were starting to have a little bit of breakthrough in the brain. So a spot in the brain starting to grow back. And what we found is that if we brought up the dose of alectinib higher than the standard dose, now to, for example, 900 milligrams twice daily, which had been tested in clinical trials, we actually, in many cases, we could induce regression again in the brain metastases. And so we had a whole series of patients like this. We published on it. And I would say for alectinib, that's quite routine for us now. If we have a patient who has CNS brain-only progression on alectinib, everything else in the body looks good, oftentimes, our first approach may be to dose escalate alectinib up to 900 milligrams. Of course, we have other options like SRS or radiation therapy, but I think if SRS isn't an option, then dose escalating alectinib can be highly effective. So Gina, it's again a long answer to your question, but I think we have flexibility. I do sometimes like to start at a slightly lower dose, especially when we have so much data on drugs to tell us that slightly lower dose is just as efficacious, but I don't start too low. And then I do sometimes dose escalate, especially if there's an issue with brain penetration, brain progression only.
Yeah, that makes complete sense. I mean, this is about personalized medicine and personalizing towards the patient. So everybody's situation might be a little bit different. So that makes
Yeah.
complete
I mean,
sense.
ideally, it'd be great if we had a blood test that could tell us the drug levels, right? And in fact, in some other cancers, for example, you may know about Gleevec in CML, chronic myelogenous leukemia, they do test drug levels. And I wish we had a way. I've had many patients over the years ask me, can we just test my level of lorlatinib to see if I'm actually in a therapeutic level? We don't have a way to do that, but I wish that we did.
Oh, somebody needs to do that for
Yeah.
us for sure. One of the questions that's coming in, and you probably have heard that a lot of ALK positive patients continue living, and a lot of us are younger, and so some are having families and things like that. I wanted to see if you could address fertility in men and women on TKIs in general.
Yeah, so it's a very complicated question. I think that ALK does play an important role in the in early neurodevelopment of a fetus. Okay, so we do feel that certainly if we would never treat, for example, a pregnant woman with an ALK inhibitor because we would worry about the impact of the ALK inhibitor on the developing fetus. Now, in terms of male and female fertility, we don't know the real impact, because as you guys probably know, on clinical trials, part of the sort of informed consent form is that you can't actually get pregnant or father a child while you're on a clinical trial. And that was because we were worried about the potential impact of any new drug on an embryo and a developing fetus. So we don't know exactly the impact on fertility, but we generally do advise the same as what was required on a trial, that you not have children while you're on an ALK inhibitor. I would say especially the mom, because of course the baby's in the mom for 9 months, and that's when they're going to be susceptible. For dads, again, the guidance is that men should also not father children while they're on an ALK inhibitor. But I can say anecdotally over the years, have had an occasional male patient who did father a child. This is when they were now on a commercial ALK inhibitor. They couldn't do that on a trial, obviously. So I know that that's possible, but again, I never had any sort of data on their fertility or anything like that. It's a very, very complicated question. Usually what we do right at the time of diagnosis, though, is if we have a young woman or a young man and we know that they are interested in a family, maybe down the road, we do have them meet with a fertility specialist right away. So that is something that you should definitely keep in mind.
Great. And we've also heard about decreasing testosterone levels.
Yes.
Are there also decreasing estrogen levels too, or do we have
Not that we've seen? And actually, the decreased testosterone levels was really seen with crizotinib. And in fact, it was primarily total testosterone levels, but not the biologically relevant free testosterone. So just to keep that in mind. So, and again, it was almost all with crizotinib and not with the other ALK inhibitors.
Oh, okay, great. Okay, so we're getting up on time here. So I really wanted to see if you could bring us all a little bit of hope and tell us about the things that you think are the most promising pipeline drugs. Then the next thing after chemotherapy, when lorlatinib fails us, what's next?
Sure. So there's a lot coming in the pipeline for ALK. And I would say starting with ALK itself. I mentioned earlier about a whole new class, 4th generation ALK targeted therapies that have really been designed specifically to overcome double or triple, or what they call compound ALK resistance mutations. And just a bit of history on this. When we were developing lorlatinib and starting to see patients develop resistance to lorlatinib, we were doing biopsies on patients, and we found that a number of the patients who had received almost all of them actually had received sequential ALK inhibitors like, crizotinib, alectinib, lorlatinib, or, alectinib, brigatinib, lorlatinib. And what we were finding is that about 20 to 25% of patients were acquiring 2 or sometimes 3 ALK resistance mutations. And we went on to show that these ALK mutations were accumulating after each ALK inhibitor. And so one after the other in sequential fashion. And at that time, this is a number of years ago, probably, I shared this early data with Jean Cui, who some of you know. She had been at Pfizer and developed, designed, made crizotinib basically. And Jean Cui had at this time started Turning Point Therapeutics. And so She's a brilliant medicinal chemist, as you guys probably know. And so she actually started designing molecules that could specifically overcome these compound mutations that we were emerging in the clinic. It was almost in real time, which is pretty amazing. And so actually, many of you know that TPX0131 has entered phase 1 clinical trials last year. That drug, again, it's a specific ALK inhibitor designed to overcome particularly compound mutations that include G1202R as one of the mutations. There's also another 4th generation drug called Nuvalent. I had also started working with Nuvalent when we were seeing these compound mutations emerge, and Nuvalent also has a 4th generation drug that should enter the clinic second half of this year. But one thing to just make note is that these new ALK inhibitors are very specifically designed for these compound ALK mutations. We know that they may have potentially less activity against certain single ALK mutations. And so I actually think their role may be somewhat limited to at least a fraction of patients, say after lorlatinib, where we have a biopsy, either liquid biopsy or a tumor biopsy that shows the presence of these compound ALK mutations. So I want just to make sure everyone knows it's not like it's going to be a new ALK inhibitor that probably will work on all patients. It really is for a subset of ALK patients after lorlatinib. Now, there are many other ways to think about targeting ALK. I know that when I spoke, I think at the ALK Summit before, we talked about PROTACs, which are proteolysis targeting chimeras, and just basically a new type of approach to targeting ALK, basically by targeting ALK for degradation within the cell. And so there are a number of PROTACs for ALK that have been described preclinically. And so that could be an approach to think about. If you could just degrade all of the ALK, that could be another way, an alternative approach to the tyrosine kinase inhibitors like alectinib and lorlatinib. I would say the challenge with these so-called PROTACs is that they still require some binding to ALK. And oftentimes the binder is a molecule like ceritinib or lorlatinib. And so again, mutations can arise that now prevent the binding. So I think degraders also are going to run up against the problem of resistance. And then the last thing I'll just mention around ALK itself is I think, John Ivredi had spoken to you guys in the past about his project and going after not ALK, but going after the partner protein EML4. And, he's working with another investigator at MGH to target a very unique approach to target the cysteines within EML4 that are required to dimerize together. So for these molecules to come together, and if you can target these cysteines and prevent the dimerization, then ALK can no longer be an active oncogene. So very, very distinct mechanism of targeting EML4 ALK. I think it's quite interesting. Again, though, at some point, if this work, progress continues on, it may still, though, run up against a problem where the cancer can mutate the cysteine that's targeted. And now, again, you've basically averted this drug. So I think these are interesting approaches to keep in mind. I think resistance is always there. And so what that automatically makes me think about is that combinations are going to be really critical, right? So that to further decrease the chance of a cancer becoming resistant, combinations can be much more able to do that than a single drug alone. And then I think beyond just thinking about how you target ALK, I do think that we really have to go beyond ALK and think about ALK inhibitors, for example, our TKIs, really as an anchor. And with that anchor, we need to add other drugs, maybe other targeted therapies, maybe other types of modalities like chemotherapy or radiation therapy, because resistance is just so complicated. And so we know we're not going to be able to prevent resistance just with these ALK TKIs. We have to be probably a little more creative, a little more ambitious, a little more risk-taking, and think about these types of combinations. So I think many of you know that we already have a number of combinations in the clinic. We've been targeting, for example, ALK and MET, because MET is known to be amplified in roughly 15 to 20% of patients after alectinib or lorlatinib, and MET can drive resistance. So we have a study combining ALK and MET inhibitors. We have a number of combinations in the clinic combining ALK inhibitors with MEK inhibitors, so trying to target downstream of ALK as well. Some of you may know that there are now 2 trials in the clinic targeting ALK and a protein called SHP2. SHP2 is an important downstream regulator and is involved in the MAP kinase pathway as well. So again, a downstream pathway of ALK. And so I think those types of combinations are really important because this MAP kinase pathway has been shown to be quite critical in terms of reactivating and driving resistance. And then there's some other combinations just to keep in mind. We and others have developed targeted therapies for a molecule called YAP, so YAP, which is a downstream transcriptional regulator in the hippo signaling pathways. And there's a lot of data, preclinical data out there now that activation of YAP can enable cancer cells to survive, even though they're being exposed to a targeted therapy like alectinib, for example, and even some data showing that YAP can drive resistance. And so something to keep an eye on. These YAP inhibitors have just entered the clinic. Novartis has one, so I should be clear about that. They're being tested currently as monotherapy, so by themselves, but the hope is if there's encouraging early data that those YAP inhibitors could be great combination partners for targeted therapies like ALK inhibitors. And then the last thing I'll just return to, Gina, is that I mentioned that besides these sort of other novel combination partners, I don't want people to forget about traditional therapies like chemotherapy and radiation therapy. We already have data that chemotherapy can combine with EGFR targeted therapies in the first line setting and improve progression-free survival and likely overall survival as well. Many of my patients also have incorporated radiation into their regimens, where we try and use radiation to eradicate any residual disease, also to treat solitary sites of recurrence. And so just to keep in mind, again, what I had said earlier about the use of orthogonal therapy, so targeted therapies and totally other types of therapies to further reduce sort of the bulk of cancer and eliminate residual disease or persisting disease.
Yeah, wow, that's a lot. I mean, really to take in to see that, actually there's a lot of options. Can you address cellular therapies like TILs and NAT and just where do you feel like those fit in with ALK?
Yeah, so I guess you're getting at sort of immunotherapies, right? How do we see immunotherapies for ALK? And as I mentioned right off, unfortunately, drugs like Keytruda and Opdivo don't work well in ALK patients. There have been many retrospective series now, and even some clinical trial data showing that ALK patients really do not respond to immunotherapies, highlighting that we need different types of approaches to activate the immune system in ALK patients. And so, Gina, you mentioned one potential approach, which is around TILs, so tumor infiltrating lymphocytes. There are a number of companies, Iovance is one of them, that has been advancing this TIL technology. And many of you know that it is quite exciting what they've seen so far, particularly in melanoma. There's a surgical specimen of the cancer that's removed, and the TILs are extracted and expanded and then reinfused into patients. And so these tumor infiltrating lymphocytes contain these killer T cells that hopefully, once they're expanded and now reinfused, can really basically exert their anti-tumor activity. And as I said, the activity that they've seen in melanoma, especially first line, has been quite impressive. And there has been data as well in non-small cell lung cancer, including I had seen some data even with EGFR patients as well, where some did have a response. But I would say overall, TIL technology is maybe not, it's still there's a lot more work to do. And my worry about ALK in particular, and I think some of you know this, is that ALK positive cancers, the problem is they tend to be what we call immune deserts. So there are just really not very many infiltrating T cells in ALK positive cancers. And this TIL technology relies on the presence of these lymphocytes in the tumor. They have to be expanded and then reinfused. And so if you really have none, then the whole manufacturing process is going to be at risk. So definitely something to keep an eye on, but I do think that is going to be potentially quite challenging for ALK, just given the baseline. Now, I think some of you know that ALK positive cancers are, it sounds funny to say this, but they're simpler than other lung cancers, right? They're not smoking induced. They don't have the genomic complexity of the typical non-small cell lung cancers. And so because of that, they're kind of quiet. They don't have lots of different appearing proteins on them to trigger an immune response. And so I think we need to understand more about the immunophenotype, the immune landscape of ALK positive tumors. And I think many of you know that Justin Gaynor and Jess Lynn are working on the Enigma project. And part of that is to profile, hopefully, as many ALK positive cancers as we can to understand what are the immune cells that are there? What is the neoantigen landscape? What are the altered proteins that are in ALK positive cancers? I think that type of work is going to be really crucial to developing sort of immune-based strategies for ALK patients.
I was wondering, just kind of when you look at all of these things together, what do you think is the thing that is going to get us past TKI resistance, and what is going to be the thing? I mean, do you think that it's even possible for us ever to make ALK positive cancer a chronic disease?
I do. And I think I mentioned earlier on that because I've had patients now who, of course, they're not the norm, but I've had patients who have been able to continue in full remission, basically on a single ALK inhibitor. It could be crizotinib from, over 10 years ago. It could be alectinib that they're on now over 8-9 years. Also, even lorlatinib, we have some patients beyond 5 years as well. And so the fact that even some ALK patients can achieve these incredible durable remissions gives me hope that, again, if we could understand the biology of everyone's ALK positive lung cancer, we would have a much better shot at basically making the disease more of a chronic condition, like you're saying. But unfortunately, monotherapy with an ALK TKI by itself for most patients is not the answer, I think, to make it this sort of long-term chronic condition. And I really think what I'm envisioning is that we really will have a shot at making ALK positive cancer chronic once we incorporate more treatment modalities beyond just the targeted therapy. And so that's why I do think I'm envisioning for the future that we got to start, I would say, with our best therapy up front. Hopefully at some point, one day, that will be actually a combination, something that, of course, hitting ALK, but also hitting the pathways that we think are most important to drive resistance or drive residual disease, which then leads to resistance. And then even after we start with their most aggressive therapy up front, we still have to come in again with some type of different therapy, an orthogonal therapy, to eliminate any residual disease, some of which we might be able to see on scans, some of which we cannot. So we may use radiation, we may need alternative medications to go after these drug persisting cells. And then the other thing I think we really need is we need to have better monitoring when patients are on ALK inhibitor therapies and even these combinations, so that at the very, very first sign that resistance is starting to emerge, rather than let it become full-blown clinical relapse, but at the very first sign, molecular detection of relapse, we add a therapy, we switch therapies, because it's always so much easier to treat early on rather than when it becomes clinically overt. So I do think, Gina, I'm really hopeful that we will get there, but it's going to be not as simple as just one therapy or one therapy after the other. It is going to be, I would say, a multimodality approach that we need to take.
That brings up a really, really great just idea. And that's really about detecting, the molecular changes that happen through circulating tumor DNA or liquid biopsy. It's really just now becoming, I think, more common that people will do liquid biopsy on progression. Can you just kind of address liquid biopsy and its potential to really detect cancer at early stages?
Yeah, so we do use liquid biopsy quite a lot these days. I would say, especially when as typical example is we have a patient in a clinic, they've been doing fine on their targeted therapy, but their latest scans show that there's likely some progression going on, say in the lung or the liver. There's some change there. And oftentimes in the old days, before we even had circulating tumor DNA, we'd be thinking about a biopsy and working with interventional radiology to perform an ultrasound guided biopsy. But these days, we do have access now to a number of different platforms to analyze circulating tumor DNA. And that oftentimes can be really revealing. If the cancer is actually shedding some of the DNA into the circulation, we can capture it and sequence it. And then sometimes that can tell us what is driving the resistance, the progression that we're seeing on scans. And so I think in that setting, again, you've now visualized the disease on scan. So it's what we kind of call macroscopic recurrence. ctDNA can be helpful. But I think what you're getting at is how could we detect it even earlier? Because as I was just saying, I think that's really the key, because we want to detect recurrence as early as possible, because I do think it's more actionable than when we have much, much less disease. And so there are more and more sensitive technologies that are being developed to try and detect, even at the tiniest quantities, that are there, potentially these mutations. And so I think there will be more and more testing of these new technologies. They likely will need to be tailored to each patient's cancer, actually, to be able to detect the cancer DNA at such low levels. But those, I think, are going to be very, very important to give us, again, the earliest view into what's evolving at the level of the cancer cell, instead of waiting just for the scans to tell us.
Exactly.
So yeah, I would keep an eye out for those new technologies.
Good. That's promising to hear that they're becoming available and being developed.
Yeah.
As far as TKI, some of us are staying on targeted therapy for years and years and years. Do you foresee any problems with that? That there may be long-term side effects of staying on a targeted therapy?
Yeah, I mean, I'm not sure how many folks are even on crizotinib at this point, but with crizotinib, we definitely do have a lot of long-term cumulative side effects. Edema, for example, is really, really common with crizotinib. And I can tell, even you can scan a waiting room and you can tell who's probably on long-term crizotinib, because you look at their ankles and they've got huge swollen ankles. So crizotinib is very well known for that as a long-term side effect. I've had patients on alectinib for many years now, and the main side effects I see kind of just, they're not progressing, they're not getting worse, but they kind of persist with alectinib long-term, is like fatigue, low grade anemia, for example, not requiring intervention, but a little bit lower than baseline, and maybe some edema and constipation with alectinib. But I have to say, overall, for the patients I've had on long-term alectinib, I've been pretty pleasantly surprised that there isn't really any accumulation or worsening of side effects during the years that patients may stay on. Lorlatinib is similar in the edema problem, in that some patients on long-term lorlatinib, not all of them, but some of them, will struggle with edema. But I feel like once patients are on any of these drugs past a couple of years, many patients, of course, you guys are so amazing that you kind of you figure out how to manage these side effects well. And so I would have to say I have many long-term patients on these targeted therapies, and pretty much what you have at a couple of years is pretty much what's going to stay with you. And with the exception of crizotinib, where the edema does tend to build.
I want to ask you a little bit about your participation with the University of Michigan project. I'm so excited because on Thursday I'm flying to Michigan and we're going to actually have cells harvested. So anyway, I'll have some Gina mice and I'm super, super excited about that. And so, like, looking at the personalization of medicine, this really brings me a lot of hope, because I'm kind of at a point where we don't have any other therapeutic options that seem to be working for me. So I just wanted to see if you could address your role in that and what you expect to happen from that project, and how that's going to help ALK positive patients.
Yes, thank you for that. So this is an incredible gift from a patient, Judith Tam, and I'm not sure if Judith is potentially on this call, so Judith Tam and the Richard Tam Foundation.
Judith, take yourself off mute and say hi.
Hi there, everyone.
Oh my gosh, there she is.
I'm really happy to be here, and it's my joy to be funding this research effort, because I have a lot of hope that it's going to make a difference, and it'll make a difference for so many.
Oh,
thank you so much.
I give you all my best, and hi, Alice. It's nice to see you again.
Great to see you. I'll just speak a little bit about this amazing gift from Judith, and this is, as you heard, it's a gift to support ALK-focused research at University of Michigan's Cancer Center. And this is a whole team of physicians and scientists and engineers and pathologists and surgeons all working together, really focused on ALK. Sophia Marajvar is the lead investigator. One of the investigators is my former mentee, Peggy Sue, who was a fellow with us at Mass General, knows everything about ALK, actually. And the overall goal, really, like I said, is to study ALK-positive cancer, to develop ways to predict the tumor sensitivity of ALK positive cancer cells to drugs, and ideally in a rapid time frame. And then, of course, most importantly, as we're studying how these cancers develop, the goal is for those investigators to develop therapies, new therapies that will work for resistance. This program will have an advisory board, a scientific advisory board to provide high-level guidance that I'm I feel so honored to be on with Shirish Gadgil, another one of the ALK investigators whom some of you may know. And this team has already been starting to not just obviously work with each other at Michigan, but actually to spread the word and to actually establish a network and connectivity with other ALK investigators, including my former team at Mass General. I just spoke to Peggy actually this past weekend, and I know that she's actually going to come to speak to you guys here, I believe in March, with other team members from Michigan. So actually they'll be able to go into much more detail about the specific lines of investigation that they're planning. But it is a really, really exciting time. And I think we're going to see a lot of progress very quickly.
We're so excited about that and that gift. So thank you so much, Judy. Thank you so much, Dr. Shaw. You have the ALK Queen on your board. So we're expecting some great things, and we can't wait. We're so excited about that. We have about 10 minutes, and I don't want to go over on time. So I wanted to see if you would address one more kind of big issue, and that's really just about what about vaccines? Can you kind of address where you think vaccines fit into our treatment, and if you think that that's something that could potentially actually be a cure?
Yeah. So vaccines are, I didn't go into them because I've been kind of racing through here, but as I was saying about sort of combination approaches, multimodality approaches, in that I mentioned targeted therapies, chemotherapy, radiation therapy, but actually immune-based therapies, which would include vaccines, I also put into that category. And so one way I'm envisioning the future is that, of course, we start with an ALK inhibitor. Like I said, that's our anchor, always the ALK inhibitor, but we build on that with, for example, another targeted therapy that shuts down the whole MAP kinase pathway. We then use, for example, radiation to eliminate any residual disease that we see on scans, or maybe we combine that with chemotherapy. We're following closely for any signs of recurrence using really highly sensitive circulating tumor DNA technologies. And we also potentially incorporate an immune-based strategy. Again, I think when we have really low levels of disease, that could be the best opportunity to then try and trigger an anti-tumor immune response. And that could very well be done, Gina, using a vaccine. There are other ways to activate the immune system as well, but since you asked about vaccine, I think many of you know that Mark Awad, again, one of my former mentees, has been working on an ALK peptide vaccine for a number of years. He's teamed up with Roberto Chiarola at Children's Hospital, as well as biotech Elicio, this company that's focused on vaccines for cancer patients. And there's a lot of preclinical work supporting the potential efficacy of these peptide vaccines. And, and these are ALK positive models, of course, mouse models. But the hope is that all that will translate into patients. And so Mark, again, they've been working very hard. He told me that they have even better potential peptide vaccines right this moment, that they're going to progress forward, and likely by end of the year, the hope is that they'll be able to submit the IND to FDA with plans to then start the clinical trial, a phase 1 trial soon after that. So hopefully we're looking at another year or so. And I think that one of the challenges here is how will we best test an ALK vaccine? Do we test in a fully resistant setting? Probably not, because I think it'll be very hard to rely on a vaccine by itself to overcome all resistance. But perhaps we test it in combination with an ALK inhibitor. Perhaps we test it even in combination with a checkpoint inhibitor like Keytruda or Opdivo, right, to further enhance the chance that you can activate the immune system. So I think there's a lot of design questions as well, but I am very hopeful about the ability to, again, if we can understand the immune landscape for ALK positive cancers, if we have a number of different shots on goal, a vaccine approach, for example, there are other approaches that we could take too, for trying to, again, perhaps engage the immune system by, for example, a bispecific type of antibody approach where you're basically bringing activated T cells closer to the cancer if you can find a ALK specific protein that you can target with this bispecific. So there are a number of different strategies I think that we can take to try and activate the immune system. And I do think that will turn out to be an important component of the multimodality approach that we take for ALK in the long run.
Well, we only have 5 minutes left, but one of the things that I asked you if it was okay if we could talk about was Linnea, because she was a hero to so many of us. And I know that as her physician, it must have been incredibly hard to lose patients. And how do you handle that? I mean, we're so thankful that you are here and you're in this space and you do this, but this must be so incredibly difficult because she was so amazing. So I just wanted you to just kind of end and just talk a little bit about Linnea.
Yeah, thank you, Gina. Yeah, I think some of you probably knew her, but some of you probably did not. And I do think, Gina, like you said, she was really amazing. I cared for her for over 12 years. And I think many of you who do know her, who read her blog, I think you know that she was, not just a lung cancer patient, she was a survivor and a passionate lung cancer activist. She liked to use the word activist. She had incredible insights and she could share them so articulately, both verbally and also in her writing. Really talented artist, some beautiful paintings. She was a devoted mom, 3 children, and she cared so much about people. That was really what's always struck me. Despite obviously Linnea had her own health issues, and yet she was always trying to help other people. Just such a kind, genuine, kind person. And I often have described her, I think even when we did the tribute at her celebration, I've described her as my hero, and I still think of her as my hero. And I think she's probably is a hero to those of you who knew her as well. Just an incredible beacon of hope and inspiration. And so, Gina, yes, her passing was really, really hard and very painful, I think, for many, many of us. The other thing I want to share about Linnea, which again, if you follow her blog, you know all this, but if you don't, just to say, Linnea was also so brave and so courageous. Back in 2008, she had no idea. I don't think she knew much about phase 1 trials. Obviously, no one knew anything about crizotinib. She was the 4th patient, ALK patient ever to go on crizotinib. But she jumped at the chance to go on this clinical trial. She volunteered basically to go on a drug that had very little basically information behind it at the time. And, she did so well on crizotinib, and this gave her courage to do many additional trials. She ended up participating in a total of 6 phase 1 trials. So think about that. She was basically pivotal to developing not just crizotinib, but also ceritinib and lorlatinib, one of the first patients on each of those drugs. And, I think she's just shared her experiences so freely. It was so, I think, helpful to the community. And I think she really showed people that phase 1 trials are really important to us. They're scientifically driven studies. They are your only access potentially to new medicines that can be transformative. And so, in addition to being my hero, I also think of her as a trailblazer, really redefining what phase 1 trials should be, and really highlighting how transformative phase 1 trials can be for patients. And so it has been hard to lose Linnea and other patients as well. Gina, I mean, this past year has been pretty hard, actually. We've lost a number of amazing patients. And I would admit that it's very easy to sometimes kind of get lost in the grief of it. But it is important to remember all the positives. And I know Linnea would want us to remember the positives. She lived with her cancer. She was diagnosed, I think, 16 years before, I think 2005 when she was first diagnosed. And during that time, Linnea lived really well. And she was able to create incredible memories with her loved ones. They have, and all of us now have, a lasting legacy of Linnea. And so I've worked hard over the years to always think about the positive and to try and channel the sadness and grief into something that's productive and meaningful, be it a new avenue of research or an opportunity to teach others, an opportunity to collaborate with others. And so I think in some ways, the loss that we all experience, that can be motivating to many of the researchers, right? It reminds us of the urgency, and it reminds us that, patients are all waiting. So hopefully, Gina, that answers your question. It's very sad, but I want to make it a positive thing, for the memory of these patients.
I think one of the things that makes you two so much alike and that made you the A team is that you, like Linnea, cares so much about the patients and about the people. And if that heart wasn't there, then I think you guys wouldn't have been such an amazing duo that you were. So with that, I'm going to allow everybody to take yourselves off mute. And please thank Dr. Shaw for coming in and spending an hour with us and giving us
Dr. Shaw.
Thank you.
Oh, hi, Frank.
Hey, how are you?
Dr. Shaw.
Thank you.
Thank you.
See you.
Thank you so much. Thank you.
Care.
Lord,
Bye,
you
everyone.
everybody.
Thank you.
Thank you so much, Dr. Shaw.
You're welcome.
Judith. Thank you. Thank you.
Thank you.
Look forward to you being in Michigan.
Me
If you want
too.
to stay on a little bit later, we have a few announcements to make, but we'll let Dr. Shaw go.
Okay. Bye, everyone.
Bye,
Have a good
dear.
evening.
Bye.
Bye.
Dr. Shaw.
What are your announcements?
Okay, I muted everyone again. So
Wow, was that not
Summer.
incredible?
that was incredible. And you did a great job. Thanks, Gina.
Yeah.
Summer.
Great job, Gina.
Unmute yourself, Summer. You got some announcements. We got 187 people still there, which is incredible.
Okay, I'll keep it brief. That was amazing. Gina, you killed it. Good job, because there were a lot of questions.
Yeah, I'm sorry I didn't get to everyone. I'm so sorry, but she was really she really needed to go right at an hour. So I'm so sorry I didn't get to all of them. But you guys
You've done
did
a
right.
great job. Great job.
Yes. And thank you, Judith. We were so happy to see you in here. Mark's text